Hello there, and welcome to our video about exciting topic of pharmacology and drug interactions. Have you ever wondered how do drugs work? Today, we're going to try to explain the complexity of drug-brain interaction and bring the light to the intriguing world of pharmacodynamics. Let's meet Sam and Lily, a pair of enthusiastic students who want to dive into the world of pharmacology and learn more about it. At first sight, you might think that drug mechanisms are super complicated, but in reality, they are wonderfully simple. It's mostly about receptors and the molecules that activate them. So the first thing that underlies any drug mechanism is a receptor. It's a specific macromolecule that is embedded in the cell wall or membrane and is involved in chemical signaling between and within cells. But to receive chemical information outside the cell, they need other molecules called ligands that will bind to receptors, activating and generating a cascade of events inside the cell. There are several examples of ligands, such as hormones, neurotransmitters, or drugs.
Now that you have learned about receptors and ligands, let's have a look at one of our example drugs, opioids. Opioids are used for pain relief and function in the brain. However, they can also trigger an exaggerated sense of well-being, which can lead to addiction. Opioids are made out of the seeds of poppy flowers or synthetically. Some of the most common opioids are oxycodone, morphine, buprenorphine, or the legal street drug heroin. But to better understand how drugs interact with our bodies and what are the mechanisms of their effect, we need to dive deeply into pharmacology. Let's have a look at pharmacodynamics and pharmacokinetics. Pharmacokinetics is Greek and comes from the word pharmakon and kineticus, which means drugs and moving or putting in motion. It is the study of how the body interacts with administered substances like drugs for the entire duration of exposure. Pharmacodynamics is Greek as well and comes from the words pharmakon and dynamicus, which means power. It is the study of a drug's molecular, biochemical, and physiologic effects or actions on a living system.
Now that Sam and Lily understand what pharmacokinetics and pharmacodynamics mean, they are wondering how we can measure and visualize this. Is there a way to see what effects a certain dose has on the pharmacokinetics and pharmacodynamics? This is where the dose-response curve comes in, a graph where the effectiveness of a substance on the receptor is plotted against the dosage. There are several effects that can be measured, starting with a neutral effect. This is when a receptor is not affected by the substance and is sometimes even inhibited in its function. The result of this is a straight line with no change of effect when the dosage increases. Next, when a substance has an effect, we can see that there is an increase of the effect on the receptor with an increase in dosage. What can also be seen is that at a certain dosage, a plateau is reached. This indicates the maximum effect a certain substance has on the receptor. Similarly, we can see that this maximum effect can also be lower, indicating that the substance has a lower effect on the receptor, leading to less activation. Lastly, the effect can also be negative. This means that the receptor performs the inverse function. With this, Sam and Lily have a very good idea of how they can measure and visualize the effects of substances on a receptor. But the professor also mentioned agonists. Now Sam and Lily wonder what the function of this is.
Unfortunately, an agonist is not like the protagonist of your current fantasy novel. However, an agonist is a drug or a substance like morphine, oxycodone, fentanyl, or heroin. These substances mimic the actions of a neurotransmitter in the brain by binding to a specific receptor, in this case an opioid receptor. The binding can cause a conformational change and activates the receptor. A great analogy to think of is a vending machine. Usually, to buy a chocolate bar, you would insert a $1 coin, and as a result, you receive your favorite chocolate bar. An agonist in this scenario would be to use a metal disc of the same size as a coin to insert into the machine, thus using the same coin slot with a mimicking coin to obtain a chocolate bar. Let's have a look at different types of agonists. There are endogenous agonists, full agonists, co-agonist, selective agonist, superagonist, irreversible agonist, and biased agonist. There are two more agonists that we want to focus on in this video. That is the partial and the inverse agonist.
Let's begin with the partial agonists. Compared to full agonists, partial agonists have a lower intrinsic activity at receptors. When binding to the receptor, they are not able to elicit the maximum possible response. But the key property of partial agonists is that they can also block the effect of a full agonist. In the absence of full agonist, partial agonists show functional agonist activity binding to the receptor to produce a response. In the presence of high concentrations of partial agonists, the action of full agonists can be reduced to the maximum response elicited by the partial agonist. For example, Think about buprenorphine, an opiate agonist that activates or occupies the mu opiate receptor. Being a partial agonist, buprenorphine does not completely bind to mu opiate receptor. As a result, buprenorphine has a ceiling effect, meaning that its effects will plateau and will not increase even with repeated dosing. Moreover, buprenorphine generates limited effect and does not produce euphoria. Thus, it does not have some of the dangerous side effects associated with other opioids.
Sam and Lily learned that agonists activate the receptor, but also some receptors can be active without an activating ligand. These receptors have constitutive activity. As a result, a new type of agonist appears, inverse agonists. These ligands produce the opposite effect of an agonist when they bind to the same receptor. When we introduce a coin, instead of a chocolate bar, this time we'll get an apple. Generally, receptors are in an inactive state, and agonists shift the activity towards the active state. This is called the two-state receptor model. If some of the receptors are in the active form in the absence of a conventional agonist, then constitutively active, inverse agonists bind and reduce the fraction of them in an active conformation. In pharmacology, intrinsic efficacy is the capacity of a drug to activate or inactivate a receptor. Agonists have positive intrinsic efficacy, therefore the ability to increase the activity of a receptor. However, inverse agonists are said to have negative intrinsic efficacy. Just as agonist intrinsic efficacy for a receptor varies with the structure of the agonist, then resulting in strong agonists and partial agonists, inverse agonists also have different degrees of negative intrinsic efficacy, resulting in strong and partial inverse agonists. This is quite relevant for the development of new drugs. With ligands that can reduce receptor activity, we can have greater degree of control over receptor function, and it is expected that this will translate into better treatment of disease.
Let's have a look at the antagonists. In pharmacology, an antagonist is not the villain of your favorite book. However, an antagonist has a similar function to the villain of a story. An antagonist stops the effect of an agonist. It binds to a specific receptor in the central or peripheral nervous system and blocks it, preventing signal transduction and therefore biological response. The most common antagonists are methylnaltrexone, naltrexone, and naloxone. They reverse the effects of common opiates and can stimulate withdrawal symptoms. So, let's look again at the vending machine. It's like jamming the machine's coin slot so that it is unable to perform its function until the blockage is removed. There are uncompetitive antagonists, silent antagonists, competitive and non-competitive antagonists. Let's have a closer look at competitive and non-competitive antagonists.
Let's start with competitive antagonists. Imagine going to the vending machine and having someone push you aside and insert a coin first and grabs your chocolate. This is similar to what competitive antagonists do. Competitive antagonists bind to the same site as agonists do. Let's take a look at the example of the mu receptor. The mu receptor consists of a binding site for opioids. However, an antagonist called naloxone, generally administered as a drug as mentioned previously, competes and binds to the same site as the opioids do, thus preventing the opioids from binding to the mu receptor. What are non-competitive antagonists then? Imagine another friend comes up to the vending machine and spoils the machine in such a manner that irrespective of how many coins you put into the machine, you never get a chocolate. Such is a non-competitive antagonist. To elaborate on this, let's look at another molecule that binds to a similar opioid receptor. It causes an irreversible conformational change. This inhibits its ability to respond to opioids. These changes can reduce the receptor's ability to activate signaling pathways effectively, diminishing or blocking the downstream effects that would normally occur upon agonist binding.
Sam and Lily now know about a lot of different types of agonists and antagonists, but they are wondering how these will affect the dose-response curve. Does an antagonist change the maximum effect, or does it change the dosage that is needed? First, there are surmountable antagonists. On the left, we can see a normal dose-response curve as we have seen before. This curve shows the normal effect of the receptor at certain dosages. However, when a surmountable antagonist is added, the curve shifts to the right. This shift is caused by the antagonist binding to the receptor and changing its effectiveness. To summarize, this means that a surmountable antagonist makes the receptor need a higher concentration to achieve the same effect. This explains what the effects of an antagonist can be, but as we also saw earlier, the maximum effect can be decreased without affecting the dosage that is needed, thus not shifting the curve to the right. To explain this, we look at insurmountable antagonists. First, we have the normal dose-response curve with the normal effect again. However, this time the curve does not shift to the right, but rather shifts its maximum effect downwards. This shift is caused by an antagonist that binds to the receptor, affecting the maximum effect of it. Therefore, an insurmountable antagonist reduces the effects of the receptor without changing the dosage that is needed. When we use our vending machine as an analogy again, we can explain this really quickly. Let's say you want to buy a chocolate bar, but all of a sudden the vending machine has 2 slots for coins. This means that 1 coin won't be enough to buy the chocolate bar. You need to put in 2 coins to get it. In this case, the extra slot for the coins is caused by the surmountable antagonist, which causes a shift to the right for the dosage, or in this case the coins needed, to get the same effect. When we look at an insurmountable antagonist, the vending machine is back to normal with 1 slot for coins. You want to buy another chocolate bar, and when you put the coin in, the machine gives you 1. However, this chocolate bar is only half. No matter how many coins you put in, you will never get a full chocolate bar from this machine. This is caused by an insurmountable antagonist being present in the machine. Because of this, the machine is incapable of delivering full chocolate bars no matter what, and thus the effect has been decreased without affecting the dosage, or in this case, the coins.
So this is our take-home message. Agonists were thought to turn on a single cellular response when binding to the receptor, thus initiating a biochemical mechanism for change within a cell. Antagonists were thought to turn off the response by blocking the receptor from the agonist. We hope that now with all this new information, you have a better understanding of drug interaction and will successfully continue your pharmacology journey. To test what you have learned today, we prepared a small quiz where you can apply your knowledge. Thank you for watching and we hope to see you soon. This video was made by Uliana, Annika, Elena, Srinidhi and Stan.